This allows calcium ions to rapidly diffuse out of the sarcoplasm of the muscle cell where it mingles with the thick and thin filaments of the sarcomeres.
There you go. Okay. So your sarcomere picture that you drew, I've got to erase this a little bit. Is basically sitting, remember it fills up, myofibrils fill up the whole cell, right? Like the whole cell is filled with myofibrils. The myofibrils are sarcomeres lined up end to end, right? Each of these is a chain of sarcomeres, another chain of sarcomeres, the whole thing is just chock full. That is all deep, so underneath all this terminal cisterna and sarcoplasmic reticulum, right? So I've got to draw it here, but understand that this whole membrane is kind of covering it, and it's releasing calcium like deep so it can actually contact the sarcomere. Okay, so I'm going to just put a sarcomere here. All right, so who's up next for our next statement?
As calcium ions are released from the SR, they bind to troponin on the thin filament, causing this protein to change shape.
Okay, so the calcium binds to troponin, which is a part of the thin filament. Okay, it causes troponin to change shape. And then what happens as a result of that, Bella?
As this protein changes shape, tropomyosin shifts over, resulting in the exposure of myosin binding sites on it.
Okay, so remember this little skinny line is our thin filament, and it's got actin, troponin, and tropomyosin on it. So let's say my hand is troponin. This hand is tropomyosin, okay? And tropomyosin's job is to cover up the myosin binding site, meaning that when troponin is in position, it is blocking myosin from attaching to the thin filament, okay? So in that little story that I was telling you about that my student Sally wrote, she talked about tropomyosin was the chaperone that nobody wanted, that kept the two away from each other, okay? So tropomyosin is blocking this binding site, which keeps the myosin and the actin separate. Okay, so read your two statements again. So start, Sarah.
As calcium ions are released from the SR, they bind to troponin on the thin filament, causing this protein to change.
So my hand here was the troponin. As it binds to calcium, it's going to change shape. And as it does, it's going to push the troponin. It's going to move it because they're all attached to each other. So troponin is here, it attaches to calcium, and when it does, it changes shape. And then Bella, what does your part say?
As this protein changes shape, tropomyosin shifts over, resulting in the exposure of myosin binding sites on.
There we go. So you have to come in and move the tropomyosin out of the way. And that movement happens when calcium binds to troponin, and troponin changes shape, okay? So that when that exposure happens, these heads, because they now are, the binding site is exposed, these heads now attach to the thin filament, onto actin in particular, okay? So they don't, it kind of looks like they grew, but it's more just that they're not disrupted, and it's almost like a magnet. As soon as that blocking tropomyosin is moved out of the way, the two are so attracted to each other, right? That's why they need a chaperone, because they just want to be together, right? So as soon as it's removed, they're going to just attach immediately. Okay, next, Sandra.
Once uncovered, the myosin head immediately attached to the myosin binding site.
Yep, there you go. So we had our troponin here. Once it has moved, you don't have to draw this, once it moved the tropomyosin, these heads immediately attach. So no effort required, magnet, like it's just gonna happen. Okay, next.
The myosin heads immediately attached to the
Okay, so we can stop there for a second. Cocked position, another word for that is high energy position. Okay, and we'll see why we call it that. Because as soon as they attach in their cocked position, what happens next?
The myosin heads immediately pivot forward, pulling the thin filament towards the center.
There you go. So these heads, both, are going to pivot inwards from both sides. They immediately attach. They're in their high energy, like spring-loaded position, and they go boom, like that. Okay, they're going to, and that, because they're attached to the actin, it slides the actin towards the middle, the thin filament, because it's coming from both sides. It attaches and it pivots. Okay? All right. So it's kind of like high energy position is you've already, like if you're doing archery, you've already done the work. So high energy is here, which means all you have to do is just let it go. So high energy is here. As soon as they are able, you let it go and they pivot. Okay? So that's the passive piece. All right, next.
This is called the power stroke. At the end of this stroke, the myosin head is left in its low energy uncocked position, and ADP and phosphate is released from the myosin.
Okay, so let me show you the low energy. I'm running out of room. So we've now gone from, oh, sorry. This is going to be our contracted sarcomere. So the heads were facing that way. They pivoted, and now we can draw it like this to show that they pulled in. Okay? So they pivoted inwards. All right, and then Cal, what's next?
I got confused here, but I thought another troponin molecule binds to the myosin head, causing it to detach from the myosin binding site on actin.
Gotcha. It's another ATP molecule.
Okay.
So what happens now, so we contracted, but the thing is that one little movement of this tiny, minuscule myosin head is not enough to make your entire muscle contract, okay? You're going to need to have a repeating process where you have myosin head, pivot, another head, pivot, and it keeps going, okay? So you have multiple heads. They're all working at alternate times so that you never just like, whoops, let it all slide back, right? So they're all grabbing on and letting go at different times. So this is one round, but we have to then be able to repeat it. So ATP binds to the myosin head and makes it detach. And then, Karen, what else do we use that ATP for?
It says the myosin head, which also acts as adipase, an enzyme catalyzes the breakdown of ATP into ATP plus phosphate plus energy.
There you go. So ATP has two roles. It detaches the myosin head from actin. And it also gets broken down. Remember we talked way back about how ATP is used, it's used to provide energy for our body to do work. So the myosin head has this double job. It not only is actually doing the pulling of the thin filament, it also is what we call an ATPase. And so ACE just means an enzyme that breaks down whatever's in front of it. So an ATPase means that it's an enzyme that breaks down ATP. So when the ATP binds to the head, it detaches it, which is important, But we also are going to break it down. And we break down ATP into what?
ADP.
What?
ADP.
ADP. So we've lost a phosphate. You've got a phosphate now by itself. And then what's the big product? Energy. And what do you think we use this energy for in this system?
Contraction.
Recocking.
There you go. To recock that myosin head. Right, so it started cocked, it immediately pivoted. ATP comes in, breaks the attachment, but we also are able to use the ATP to recock the head. As long as the binding site is exposed, what will happen to that head? It'll reattach and immediately pivot. ATP breaks it, we use it energy to recock. It immediately attaches, pivot, right? This whole cycle. And that's happening on like hundreds of heads on this thick filament in thousands of sarcomeres, in thousands of cells all at once. Okay? All right, what's our next? Let's finish our little paragraphs and then I think we hit the end.
So as long as calcium ions are present in the sarcoplasm, these steps would.
Yep. So as long as calcium is there, why do we depend on calcium to be what decides if this happens or not?
Because it changes the shape of troponin, which uncovers the myosin binding heads because it moves the tryptomyosin.
There you go. So as long as calcium is there, the myosin binding site on actin is exposed. And as long as that's there, and as long as we have enough what?
Calcium?
Enough calcium and enough ATP. This process will just go and go and go, okay? What do you think? What if your brain says, stop contracting? What's gonna have to happen? Amy?
Close the gate.
This message stops arriving here, right? So now the positive just stays out there. What happens to your voltage gated calcium channel? Closes. What happens to exocytosis of ACH? Stops. What happens to chemically gated sodium channels? Close. Voltage gated sodium channels? Close. All these voltage gated sodium channels? Close. Are we opening up the calcium, voltage gated calcium channels anymore? Nope, they close. So we're no longer depositing calcium, which means we no longer have something attached to troponin. Tropomyosin slides back into place. The myosin heads let go, and everything goes back. Okay? It's an amazing thing.
How do they figure this out? There's gotta be a lot of guessing, right?
Well, there's a lot of guessing, right? To start, it's making a hypothesis in research.
And they verified it all?
Oh, yeah.
It's just crazy.
Isn't it wild? So there's some videos that show like animations of what's happening. And there's one that actually has these like myosin heads, like you can see the molecules doing their job. It's amazing stuff. And what you do is like you figure these things out and then you go in and you say, okay, I'm gonna create this drug, in the lab that's going to like maybe prevent the opening of this, or it's going to bind to acetylcholine so it can't be released. And I'm gonna see what happens. So you interject some kind of kink in the system based on your research that you're working on. And then you see, if I insert this into that body, into that test animal, whatever it is, what's gonna happen? And then you see that nothing happens, right? So you at least see, well, what I am hypothesizing is at least somehow involved. I might not know exactly how, like in the details, but I can tell that it has interrupted the system when I messed with that piece, and you just start digging in deeper and deeper with all of that.